Okay, so I'm going to be talking about the resting membrane potential of neurons. So all living cells are negatively charged inside the cell relative to outside of the cell. And in neurons that are at rest, which means that they are not currently actively engaged in either generating or sending electrical signals, this generally means that they are somewhere between 60 to 90 millivolts negative inside of the neuron relative to outside of the neuron. And every neuron is going to have its own resting membrane potential. Okay, so there's going to be a unique value or number to that resting membrane potential for each neuron, and it's going to be somewhere probably right around in that range. So for the purposes of our little demonstration here for this neuron, we'll give it a nice kind of middle value there, and we'll say that the resting membrane potential for our neuron is, let's say, somewhere around negative 75 millivolts. Okay? Now, remember that the cytosol, which is the fluid inside of the cell, as well as the extracellular fluid, which is on the outside of the cell, has lots and lots and lots of different types of ions dissolved in it. So there are things like sodium ions, okay? And there are going to be lots of sodium ions. We'll just make lots of little red dots to represent our sodium ions. So there's lots of sodium ions all over, dissolved outside in the extracellular solution. And there's some, but fewer, dissolved inside in the cytosol. Okay? And there's also potassium ions. Okay? And there's a lot more potassium inside. So if my little blue dots here are my potassium ions, we have more of them inside relative to outside. And there are also going to be, say, chloride ions. And there's going to be more of them outside. There are going to be calcium ions, magnesium ions. Okay, so again, just all sorts of different types of ions dissolved in those two solutions. Okay, and the point of this really is that if you look at this, there's a lot going on, right? We've got lots and lots and lots of different types of ions dissolved inside and outside, and there are going to be different concentrations of each ion inside and outside. So certain types of ions, like for example, potassium, are going to be more common inside of the cell, and other types of ions are going to be more common outside of the cell. So we've got two really, really, really different solutions with really different ionic compositions inside and outside, and they're kept separate and kept distinct from each other by this cell membrane. Okay? And out of all these different types of ions, and these are just a few of the different types of ions that are in these solutions, the one that really plays the biggest role in terms of developing and setting the value of the resting membrane potential for the cells is this one right here, potassium. Okay? Potassium is the major player in developing the resting membrane potential in the neuron. Okay, so why potassium? Right? All this stuff going on, all these different ions. What is so special about potassium? Okay, so there are two things that are really fundamental or key or crucial for the development of the resting membrane potential. And one of them is the fact that we have a large potassium ion gradient across our membrane. And in the case of potassium, this means we have a lot more inside the cell relative to outside. Okay, so far so good. But we have lots of different ions that have concentration gradients inside and outside of the cell. So that's not really unique to potassium. The other thing, and this actually is really key to this whole process, is the fact that unlike the other ions that are present, potassium is able to cross the cell membrane while the cell is at rest, while that neuron is in its resting state. And that really isn't true, at least to the same extent, for all of our other ions there. Okay, so let's go back just a minute and talk about how ions get across a cell membrane. So, ions are charged by definition, okay, which means that they're hydrophilic, which means that they can't cross this lipid bilayer cell membrane all by themselves. They can't directly move across that lipid layer because of their charge, because of their hydrophilic properties. So in order to get across, they need some help. And they need help from specialized membrane proteins called ion channels, right? So if we just draw a little protein in there, okay, that forms a potassium channel. Now, our potassium ions are able to move across, going either in or out across that membrane through this protein that forms a pore in the membrane. Okay, it's kind of like how I can't walk through walls. I need an open door in order to get in or out of a room, right? So ion channels are like little protein doors that ions are able to go through. If there isn't a door there or the door's shut, I'm stuck inside the room or outside of the room. Likewise, if there isn't an ion channel there or that ion channel is closed, the ions aren't able to cross. And there's specific types of ion channels that will only let in or out certain types of ions. So for example, sodium ions go through sodium channels, potassium ions go through potassium channels, chloride ions go through chloride channels, etc. Okay? So what makes potassium special in this case is the fact that there are a certain type of potassium channels in the neuronal membrane, and we call these potassium leak channels. And these type of potassium channels are open all the time. So even when that cell is at rest, potassium is able to get across the membrane, and there's always some potassium permeability. And again, that's not really the case, or at least not to the same extent, with a lot of these other ions. Okay, so here's our cell. And in this case, I erased all of the other ions just for simplicity's sake. So all we're looking at here is the intracellular and extracellular concentration of potassium ions. But remember, there are still all those other types of ions and all those other charges that are still here. We just simplified it down so we could really see what's happening with the potassium ions. Okay? So we've got a lot of potassium inside. We've got not quite so much potassium outside, right? And we know that this membrane that separates the cytosol from the extracellular fluid is permeable to potassium because we have those potassium leak channels that are open in our resting neuron, okay? So clearly we have a large concentration gradient for potassium. And since we know our membrane's permeable, we know that our potassium ions are going to begin to diffuse out of the cell down their concentration gradient. Okay? So far, so good. But remember, potassium ions are charged. Okay? They're monovalent cations. So as they start to leave down their concentration gradient, they're going to bring their positive charges with them. Okay? So as they leave, we're going to see the inside of that membrane start to get a little bit more negative, and we're going to see the outside start to get a little bit more positive. All right? And the other thing to remember, so if we just remind ourselves that each of these little potassium ions has a positive charge associated with it. Okay. And we know that opposite charges attract, right? So as the inside of this neuron becomes a little bit more negative relative to the outside, the potassium ions are going to become less and less likely to leave because they're going to be more and more attracted to the negative inside of the cell. So what ends up happening is by diffusing down its own concentration gradient, potassium starts to create an electrical gradient that goes the other direction, that's tending to hold the potassium inside of that increasingly negative interior of the membrane. Okay, so if this is our concentration gradient going outward, we now have, we're starting to develop an electrical gradient for potassium that's going inward, that's tending to keep those potassiums inside. And the more potassium goes out, the larger that gradient becomes, okay, until finally we get to a point where the gradient going outward, that concentration gradient outward, is equal but opposite to the electrical gradient that's going inward. So the tendency for potassium to leave becomes exactly balanced out by the tendency for potassium to go inward down its electrical gradient. Okay? And at that point, if you consider both of these together, what we end up with, oops, make that look a little bit more like an arrowhead. Or my little two-headed arrow here. Okay, we reach a point where that electrical gradient becomes big enough, right, that we hit a situation where there's no net movement of potassium in either direction. And that is called the electrochemical equilibrium. Okay? So notice we still have a concentration gradient for potassium. We still have an electrical gradient for potassium. But since those two gradients, those two forces, are opposite each other and equal to each other, we have equilibrium of that entire system, and there's going to be no more net movement of potassium out of the cell. And that electrical gradient, that separation of charge across the membrane, is going to stabilize. And at that point where that electrical gradient stabilizes, we have now developed our resting membrane potential. So that really is what the resting membrane potential is. It is the voltage across the membrane, that electrical gradient that has developed at equilibrium because of the movement of potassium down its concentration gradient. Okay, and there are a couple things now that are really important to point out with all of this. And one is during this process, the concentrations of potassium inside and outside of the cell really don't change significantly at all. It takes very, very few ions moving out across the membrane to change that electrical gradient enough to hit electrochemical equilibrium and to create the potential. In fact, it's been calculated at something like 1/10,000th of the potassium ions actually have to leave before we hit electrochemical equilibrium. So for all intents and purposes, if you look at it practically, the gradients really have not changed chemically at all, and that's an important point to bring up. The other thing I want to make sure that I say is that yes, other ions do contribute to this, but to a very, very small degree. And again, that really has to do with the fact that at rest, Okay? The cell membrane is predominantly permeable to potassium as compared to those other ions. And if those ions can't move across their concentration gradient, they're not really going to contribute to the development of that electrical gradient. So they don't really play a role. So predominantly, it is all about the potassium. Okay? Sodium, again, it contributes a little bit, but at rest, even though there's sodium channels in the membrane, most of them are closed. So sodium really isn't going to contribute much. And then the last thing is that in different types of cells, different ions can contribute more or less. It really depends on the particular concentration gradients in that cell. It depends on the types of ion channels that are open at rest, and it's going to be variable. For example, chloride. Chloride does not play much of a role at all in the development of the resting membrane potential in neurons, but in skeletal muscle, it plays a huge role. In fact, in human skeletal muscle, chloride ions may even be more important in establishing the resting membrane potential than potassium ions are. So it's going to be variable. But even though the specifics are different in muscles versus neurons or in different types of cells, the underlying point, the underlying mechanism for how that membrane potential develops is the same. Okay? In all cases, it is the membrane potential is the voltage across the membrane. It's the electrical gradient across the membrane at electrochemical equilibrium. It's that separation in charge that is there when the system reaches equilibrium and that electrical gradient stabilizes.